the enzymes that actually drive these reactions, these transformations, go to the unique genomics of individual bacterial strains. And as people have probed that genetic pool and looked for the enzymes that are responsible for those transformations, they find more and more organisms that were never known before that have that enzyme function or that complexity, and that have the ability to interact synergistically with other organisms. So they almost gang up on transforming nitrogen. And so it's not nearly as straightforward as what I'm going to present here as the first round of this. But Leon really has this under his belt and is going to give you all the new stuff, the current depth of understanding. And fortunately, the learning part of that certainly has occurred right here within TRA. And your exploitation of the discovery and the knowledge and the power, say, of CoMaMox, and what its tolerance is for a different oxygen profile, has led to a very creative aeration basin control strategy that's paid back millions of dollars by understanding how to cultivate and select for a particular microorganism subspecies, right? So there's, this is kind of geeky stuff and academically interesting when we're talking about enzymes and functions at the cell membrane level. But it should always come down to a very practical, why in the heck does all of that stuff matter? Well, here at Cruise, it matters because now you have designed a system with that in mind, and it's saving you millions of dollars, and you're achieving an effluent quality that's reliable and stable at a much, much, much lower cost. That's what sustainability looks like in the era now that we're living in. And this is in the hands of operators now to impact the world that way. Yeah, it's same stuff, different day, feels like it, but there's something really, really special going on underneath the hood of the car in terms of that microbiology that's doing nitrogen transformation. So I'm going to give you the basics, and then I'm going to let work by others once I'm finished and out of your hair, finish the story. The nitrogen cycle then is the basis. What we've got here on the slide is really the basis for what we're trying to understand in nitrogen transformations. Of course, atmospheric nitrogen, what we're breathing, you know, the atmosphere that we breathe is what, 79% nitrogen gas. The rest of it's oxygen with a little bit of other things. But essentially, it's about high 70s% nitrogen, and that nitrogen is a pretty inert gas. So it has to get fixed. Somehow it has to get from the atmosphere into the food chain so that we can actually utilize, because remember yesterday we talked about that nitrogen atom that lives up in the atmosphere that's stuck as an inert molecule, somehow has to get made available to become part of our proteins, right? Every protein is built on amino acids, and every amino acid has an amine group, which is a nitrogen atom stuck on it. You don't get a protein without having a nitrogen. You don't build a DNA sequence without having a nitrogen, right? So there's the nitrogen has to somehow get into the food chain so that those of us who need organic nitrogen or inorganic nitrogen that is not in this inert form can incorporate it, right? So nitrogen gets fixed. So nitrogen fixation actually creates organic nitrogen, and typically that's happening in the plant world. There's also nitrogen fixation occurring at the bacterial level in ponds and in the ocean with blue-green algae, different single-celled microorganisms have the ability to actually take in nitrogen as a gas, and they have, guess what, enzymes then that take that nitrogen apart and stick it into organically bound forms, then that something else can consume. If it's grass we're growing that then contains that organic nitrogen, the cow comes along and eats it, and now that nitrogen gets incorporated into the cow, and we can consume the cow. But the point is, we're moving it from the atmosphere into the food chain. So we get organic nitrogen in. That's the form that we're typically dealing with it when we're dealing with it in a wastewater context. And that organic nitrogen then largely degrades to ammonium through bacterial hydrolysis actions. So bacteria will degrade that. And that ammonium then in the wastewater context is very bioavailable for transformation by nitrifiers. And remember, the bacteria are looking for something that gives them a reason for life, and that's about the energy that they get. And organisms that can get energy out of a compound will do so, and there's energy held within the chemical structure of ammonium that can be released by oxidation, and that's what the nitrification reaction is all about. It's actually an energy yielding mechanism for bacteria of different strains. So you take ammonium, it gets oxidized, and it goes through two oxidation states. The first release of energy or oxidation state is nitrite, NO2, and then the second oxidation state is nitrate. Okay, so the conversion of ammonium from ammonium to nitrite to nitrate is called nitrification. These are inorganic now because they've been freed up from the carbon atom that they were initially attached to. If it's organically bound nitrogen, it's tied up with a carbon atom. But if it's once it's in an inorganic form, then it goes through nitrification. How many of you have had little kids and wet diapers in your life? Is that a pretty good number? So you take a wet diaper, you throw it in the diaper pail. Sits there for a while. You pull the lid off the diaper pail, and there's a lot of odors that might emanate, but what's a typical odor that comes from a wet diaper after it's been sitting for a while? Ammonia. Where's the ammonia come from? Because that's exactly what we're looking at here on that part of the cycle, ammonia. Well, one of the principal ingredients of urine is urea. Urea is a carbon atom with a couple of amines stuck on the side of it. And urea is very readily hydrolyzed by bacteria with an enzyme called urease. Okay, so those bacteria are indigenous to the flora of wet diapers. And so it's almost an automatic reaction where you're seeing organically bound nitrogen, which is in the urine, readily decomposed to free up the ammonium just by the bacteria deaminating that organically bound. So the point is, there's conversions that are going on, that ammonium then is the starting spot for nitrification. Once we get to nitrate, then that nitrate is available for other purposes. Now, nitrate can be utilized and taken up by plants and put right back into an organic form. If you've ever done any ag work, we're fertilizing fields, we might feed something like UA and urea ammonium nitrate, typical ag fertilizer. Well, the urea has available nitrogen, ammonium has available nitrogen, and the nitrate, actually, when we put it on the soil, is actually reduced to ammonium by soil microorganisms. And so there is a pathway for the nitrate to actually be taken up and utilized. But in the wastewater context, largely what we're dealing with is when nitrate is formed, we are trying to get that to go back to the atmosphere, to actually get that nitrogen out of the water. We don't want it continuing to be a biological impact. And if we can put it back into the atmosphere from whence it came, that's what the atmosphere is saying, give me your nitrogen. This is not greenhouse gas problems, it's where the nitrogen belongs. It's the nitrogen balance, right? So the denitrification, D is from the Latin that means from. So nitrification is one direction, denitrification goes from that or backwards. So nitrification is an oxidation, denitrification is going away from that, which is a reduction. So we're reducing the nitrate under now what we're calling, we use this term yesterday, anoxic conditions. It's anoxic by definition because nitrate or nitrite is present. So under anoxic conditions, then that nitrate serves a very useful purpose for organisms. I really liked it. Serves a very useful purpose to the bacteria because remember we talked yesterday about the engine burning the fuel, and if we choke it and we don't have enough oxygen to oxidize the fuel, if there's a carbon source and we don't have enough oxygen, we run oxygen starved, and there's the demand for oxygen is not being met. Okay. An alternative biological source of oxygen, if it can't get free O2, remember on that electron tower, the next thing down that is useful to the bacteria is nitrate. So they'll use the oxidizing power of nitrate. It's like opening the choke on the engine. Instead of oxygen flowing through the carburetor, we got nitrate flowing through it. But the oxidation step happens the same way to allow that carbon to be combusted. And if we look at where that nitrate fits actually in the electron transport chain, at the terminal end, when electrons move along in the electron transport chain, where ATP is actually produced, nitrate slips right in place of where oxygen would have been if it could have. And so the switching from aerobic metabolism using oxygen as a final electron acceptor to nitrate as a final acceptor, electron acceptor, it's almost immediate. It happens very, very, very quickly. So it's a natural act for heterotrophic bacteria. Heterotrophs are the organic loving bugs. It's a very natural act then for them to, they're wanting to oxidize carbon to get their energy. And they just as soon use oxygen, but if that's not there, they take nitrate, right? So nitrate reduction can go back through nitrite and back to the atmosphere, and we're good to go. So that's kind of a very lengthy explanation kind of the nitrogen cycle. You'll note that now we have, I've got Animox that we've added in here that says, hey, there's a, there's some other reactions there's other bugs than what just did this simple pathway. The Animox stands for anaerobic ammonia oxidation. Well, there are species now that, and Leon will talk about these, but this is a shortcut, if you will, in the nitrogen cycle that allows ammonium to be oxidized anaerobically. So the simple nitrogen cycle that we started with years ago in the textbooks is becoming more and more complicated as they're probing and finding more organisms with more capabilities and more overlaps and interconnects. So it's admittedly getting more complicated, but within that complication is coming technology that's going to be awesome. Because if we can engineer then a system to anaerobically oxidize ammonia, we save a whole lot of oxygen costs right there, right? And so there's, Leon, you've got the right guy coming up as the cleanup hitter to go through the new technologies then that are exploiting these new microbial fractions. So that's my my opening comments about that. So when I was trying to learn myself about nitrogen, I was a little bit confused by all the different terms that were applied. So this next graphic was simply my attempt to try to take the nitrogen terminology and get it out there so that we can see how things lay out. So we start with, if you hear the term, nitrogen can come in different forms. One is an organic form, and the other, well, and within the organic form, and organic means it's tied up on a carbon atom, so the nitrogen is attached to a carbon atom. It can be further subdivided then into a particulate or a soluble form. A particulate is simply a large enough molecule that it's retained on a filter paper of a certain pore size, right? So it's the chunks that are organically bound versus the soluble. Okay, so we got soluble and particulate organic. If it's not organic, then it's inorganic. And inorganic nitrogen comes in three flavors: ammonium, which is the kind of the big dog on the porch; nitrite, which is an intermediate stage of the oxidized form; and nitrate. Okay, so if we talk about organic nitrogen, it's those pieces. Inorganic nitrogen are these pieces. Some regulatory permits are limiting total nitrogen discharge. Some are limiting ammonia discharge. Some regulatory permits are limiting the TIN. You'll get a TIN limit. A TIN stands for total inorganic nitrogen. So it would be the sum of all those three pieces, right? So there's one of the reasons for just kind of learning the acronyms in the alphabet soup here is just to make sense of what is being asked of us by our community or our regulators, of what species of nitrogen is of concern, because we target those to remove the ones that we're required to. Okay. If you hear the term TKN, which is it's likely if you go very far in your reading or learning of nitrogen, you're going to run into TKN. TKN stands for total Kjeldahl nitrogen. The Kjeldahl process, it's a laboratory procedure. And the total Kjeldahl nitrogen represents the organically bound nitrogen plus ammonium. Because the way the Kjeldahl test is done, it actually takes the sample of wastewater and it digests it so that it releases all of the organically bound nitrogen as ammonium. Okay, so during the digestion of the Kjeldahl method, it's converting everything into an ammonium form. And so Kjeldahl nitrogen is expressed as it's measured as ammonium. So any ammonium that's there is actually adding in. So TKN would be ammonium plus organically bound. Okay, so it actually includes this little piece of inorganic nitrogen, but that's the TKN. Soluble Kjeldahl means that we simply filtered the sample and got the chunks out before we digested it. Okay. If you see the term NOx, this is referring to the oxidized forms of nitrogen. So it would be the nitrite and the nitrate, okay, the NOx. Total inorganic nitrogen, which is a regulatory standard in some states, is referred to as TIN. Total soluble nitrogen would be everything that passes through a filter paper, okay? Everything but the chunks, organic and inorganic. And then total nitrogen is the whole enchilada, right? So this was, I hope that was useful to kind of just spread these pieces out and look at how that terminology applies to the whole pie. We try to understand, when we're talking about nitrogen removal, kind of what the fate and transport is of the nitrogen, because there's a lot of attention now certainly being given to the importance of transforming and removing a certain part of the nitrogen to prevent then the downstream of impacts from eutrophication and so on. There's a bunch of places that the nitrogen can go. One is it may come in and go out. That's what we just call a pass-through. Okay, that would be a fate. It came in, it's going right on through. Okay, Volatilization is referring to the fact that some of the nitrogen might actually just leave by being stripped. And in fact, I can remember back in my early days of learning about advanced wastewater treatment, that the South Lake Tahoe plant was a test site where a lot of advanced treatment technologies were being trialed full scale. And they had ammonia stripping there. And what, in that case, what they would do is raise the pH of the primary effluent very, very high, and then put it across like a cooling tower or with fans blowing air, counter current. And when you raise the pH of your wastewater, ammonium, because of the equilibrium shift, goes from the soluble to the gas phase as a function of pHs. Some of you are probably very well aware of that. But if not, the higher you raise the pH, the more the equilibrium shifts and the ammonium wants to be in the gas phase rather than the soluble phase. And once you get up to a pH of about 11 or so, almost all of the ammonium is available to be stripped if you can just blow air through it. And so one way of removing the ammonia is just raise the pH and blow air through it and strip it out and put it right back into the atmosphere. Okay. That isn't very widely practiced anymore, but you'll note, I think if you do pH, like line stabilization of sludges or cases where you've really jacked the pH up, you may see ammonia really coming off, and that's simply because the pH is changing the equilibrium. So ammonium will stay in what we call the ionized condition at neutral to low pH, and it will be in the unionized condition, or turning to a gas, at higher pH. There's some relevance to knowing that also if you're doing wet testing, whole effluent toxicity testing. We have a lot of responsibilities as operators, and another one of those is what we discharge actually doesn't kill things. Not only does it have to be low in DOD and TSS and coliforms and everything else, but it also has to be tolerable to fish and maybe other aquatic test species, whether it's a fathead minnow or a daphnia or a Cereodaphnia or a kind of, you name it. It depends on the locale and what they're choosing as their array of test species. But a very simple way to kill a fish in a toxicity test is to have excessive ammonia and have a high pH. You can, an ammonia level of, simple ammonia level of 1 to 2 ppm, which is not very high in the final effluent, will kill trout in a toxicity test if the pH gets up above about 8.5. Rainbow trout are not our test species here in Texas, thank goodness. I think we've got fathead minnows probably. I don't know if you could kill a flathead minnow with a sledgehammer, but no, I know you can. Yeah, they're a legitimate test species, but trout are particularly sensitive in certain parts of the world, particularly those where I spend a fair amount of my time. It is the test species, rainbow trout, so we've become quite familiar with ammonia toxicity, and we're sensitive to that. In any event, volatilization then is one place where ammonia may go. So if you're trying to do a ammonia balance on a system where you're tracking how much ammonia came in and how much was removed by different mechanisms, that would be one part that may account for the failure to be able to close a mass balance around the ammonia. It could be some volatilization happening where it's simply, like my kids would say, Dad, it just disappeared. It just disappeared. Assimilation, of course, we made a big deal about in this course. Assimilation means that the nitrogen is very important to the cell and is a part of the cell structure. So some of the nitrogen is simply going into cellular mass and becoming part of that biomass. Remember yesterday, so somebody keep track of which one goes first and second, because we're going to come back to these. I think that was first, that was second. Yesterday, I think we said that the average bacterial cell is somewhere around 10% nitrogen on a dry weight basis. So, it's 50, you weigh these boogers and you look at the chemical composition. They're about 50% carbon, about 10% nitrogen, and about 1% phosphorus, unless you've got PAOs, or the phosphorus accumulators, which might be 5 to 10% phosphorus. So there's a good amount of nitrogen that's simply being picked up and tucked away in the cells. Another fate then would be being oxidized. It's actually not a cellular component that's building a protein or a piece of DNA or nucleic acid or whatever. It's actually a food source. It can be oxidized. So that's nitrification, where it's really a energy fuel for the bacteria. If it's nitrified and you've invested money putting oxygen atoms onto that nitrogen, we pay a lot with our aeration to take that reduced nitrogen form, which is ammonium is in a reduced form, and we got to stuff oxygen on there to make it an NO2 or an NO3. To drive that oxygen on there and lock that up takes a lot of energy. Now, of course, it's being driven in and locked up by the enzymatic effort of the bacteria, but the energy to build those enzymes and to make those chemical bonds happen, snap together. It's kind of snapping together a little bit of a Lego set here, molecularly. That is an energy demanding process, that oxidation process. Once you've got that oxygen tied up there and there's oxidizing power now that we've created, that oxidizing power can be made bioavailable to do denitrification. And heterotrophic bacteria like to find some alternative source of oxygen. If they can't get it from your blower or your surface aerator, the next place they're going to look is, hey, you got any nitrate to spare? And if it's around, they'll switch to that very, very quickly. So it can be reduced. And when it's reduced, typically it's going right off to the atmosphere because it volatilizes. And then the final fate that I wanted to have you consider, certainly, and this has been relevant at cruise, it's not relevant if you're using other disinfection methods, UV, ozone, not impacted by the nitrification process, but certainly if we're using chlorine as a disinfectant, then the formation of combined forms of chlorine to establish a chlorine residual is impaired by the presence of the partially oxidized nitrogen of nitrite. And remember, the term that we used for that yesterday was nitrite lock. Very likely you might see that on the certification test, folks. So, hopefully that's an easy point. We want all the gimmies we could get. So nitrite lock then is a phenomenon that you guys have lived through. You felt the pain and suffering and the cost of that. About 5 parts of chlorine are required per part of nitrite that is there. And so being kind of stuck in this partial nitrification, where you have some buildup of nitrite, is going to come at a cost of chlorine demand if that is your disinfection method. So we really are trying to drive all the way through to get complete oxidation to avoid that nitrite lock situation. Ammonium that slips through is very good and reactive to hypochlorite. So one of the techniques that we try to do, if we've got some nitrite demand that's robbing us of getting an effective chlorine residual, we'll sometimes try to slip a little bit of ammonia into the effluent, because the ammonia is also very attractive to the chlorine, and it'll form chloramine. And it kind of hides the chlorine in a biocidal form from the nitrite. There's a chemical competition going on there. So it is a little bit of a sleight of hand or trick, if you will, to reduce the impact of the partial nitrification of the nitrite lock and the chlorine demand. So, kind of a lot to say on the fate and transport there. So we got to understand kind of what our goal is and how do we get there. I'm going to turn the lights down now so that those who are waiting for your morning nap can uh, can get into the proper depth of sleep. I've been trying to learn more about sleep cycles, right? So, there's that very deep sleep, and there's that kind of that REM condition, that, and a couple of places in between. So, if you're needing some of that rehabilitative sleep, now's the time to get it. But I'm going to show some micrographs, so I need the screen a bit more visible for that purpose. So what is our goal with, with the nitrogen, and, how do we get there? We're going to be looking at the biological transformation. We always are dealing with a mixed culture. I think that's probably very obvious from yesterday, looking at the numbers and the species and the diversity and the complexity of flock. This is a very mixed culture. Where are the nitrifiers? Where are the denitrifiers, and how do you find them? There's different ways that we've gone about this. I can tell you that when I came across the plains on the Oregon Trail in a covered wagon with my old microscope, and that's what we had in the day, what we'd call the old school, there are some particular morphologies, meaning shapes, that we've at least told the story, and we think it's true, but there's particular morphologies that really look like nitrifiers. And I've learned this at the feet of David Jenkins, Mike Richard, and others. And I think they are very, very credible people, and they would say that they were with fairly high certainty could look at flocks and say, now that morphology, that piece right there, those are your nitrifiers. So I drank the Kool-Aid, and I think I didn't die from it. I think it's largely fairly accurate. And to this day, through phase contrast microscopy, and just looking at a lot of them, and looking at the data from those plants, from which we can infer that nitrification was happening. We can prove that we know that nitrification had to happen because we see the nitrate is present, right? So we know something had to produce it, and we always keep finding these same particular clusters when we see that thing happening. So we've drawn these kind of connections. We kind of connected the dots. So there is a school of us, old dogs, that say, phase contrast microscopy might be one way to see the nitrifiers and reveal that. I'm going to also admit that it's hit and miss. There are certainly nitrifiers that you'll never see definitively in the microscope. But there's some times when, if I could bet a fair amount of money on it, I'd put the money on what I think I'm calling would be right. But as the world of microbiology has advanced, along has come much more precise ways of targeting the organisms that are actually doing that job. And so there's a fluorescent tagging technique, this fish probing. It's fluorescent in situ hybridization. A lot big words for basically the idea that scientists much brighter than me have developed fluorescent tagging or dyes that are attached to molecules that specifically react with a particular part of the cell where the enzymes that the nitrifier is producing. Okay, so it's basically you add this reagent that is fluorescent, and it goes in and it ties up with a bug that has that particular enzyme that does that. It might be AMO, ammonium monooxygenase, or it may be HAO, but there are specific enzymes then for these steps, depending on which step of the nitrification process is going on. So they know the enzymes, they know how to tag them, so that's very cool. So they do fish probing. There's also then genomic analysis where they can go in and actually look at the genetic sequence and based on the genetic fingerprint, know that these genes code for genes code for the manufacturing of proteins, and the proteins are what is what the enzymes then get made from. So they basically can look at the genetic sequence, and based on the particular genetic sequence, tell you what that organism is capable of doing. So this has really improved immensely then the depth of understanding of who's doing what and who gets credit for what. And it's really changed the way the books are being written. All right. Furthermore, how do you find them? Well, we can just look for the trail of breadcrumbs that they leave through the forest when they walk to Hansel and Gretel's house, right? In other words, we're looking for the TKN, the ammonia, the nitrite, and the nitrate. And from that, it's indirect evidence. We're inferring that they're there, okay? It's inferring the presence of nitrate. So this is what this is. These are just taken out of published papers that show the fish technique. These are different samples, but basically I've color-coded this here where anything that takes up the red fluorescent dye is a heterotroph. The heterotrophs are what's eating our BOD. Those are the carbon-loving bugs, okay? The green bugs, the green fluorescent tags, I tag right to the ammonia oxidizers, ammonia AOBs, that's taking the first step of nitrification from ammonium to nitrite. The aqua colored tags go to the NOBs, the nitrite oxidizing bacteria. That's the second stage in the Texas two-step, right? You go, first step is ammonium to nitrite, second step is nitrite to nitrate, do-si-do, and around you go. You know, that's the typical standard kind of two-step reaction. Now, it turns out, of course, that it's not quite so tidy because they have now found organisms that do the whole thing. And that's the Comamox, the complete ammonia oxidizer, has the enzymes for the whole, to do to both of those in one cellular structure. And then the yellow fluorescent tags here are those that are denitrifiers. So what it does is it allows us to say, let's look at the floxins, and we can see that we do have, we've got heterotrophs, we've got nitrite oxidizing bacteria, we've got ammonia oxidizing bacteria, we've got denitrifiers, and it's a much more visual and frankly, precise way to prove presence or absence. Okay. TRA, with Leon's guidance, has done a bit of this, and this is from data from a few years back, basically where it looked at AOB and NOB distribution in your flock. They were looking for what kind of spatial distribution might be. You know, we're thinking, well, maybe the nitrifiers would be on the outside of the flock and whatever. It turns out that they're pretty widely dispersed. The white, this DAPI tagging is the other heterotrophic organisms that are in there. So those are the carbon lovers. But you can see that certainly we've got both AOBs, NOBs, and heterotrophs represented there. No surprise. The DNA analysis is really advancing, and is simply using DNA technology then to establish very precise identity and fingerprinting. So you guys have done a lot of that as well. So the techniques are out there. Of course, the genomic testing and typically the fish probing is something that you contract with folks in a analytical lab offsite to do. There's very few folks that would have that in-house capability. I've seen a rare occasion where folks have tried to do some in-house fish probing, but this really belongs in academia. Utilize the resources of research institutions for that expertise, and you're far better off. All right. The simple way to teach nitrification, and I'm going to say that this is oversimplified with some intention, is to say that we start with ammonium. Okay. Now I'm showing it as an ion, NH4 plus, because it's in a dissolved form at neutral pH. If you prefer to write it as NH3, that's fine. I do not give a rip about balancing chemical equations or getting them perfectly precise, but I like to show it as the ion here. But ammonium converts to nitrite and nitrate. That's the full complete oxidation of ammonium. I'm showing you a couple of species, Nitrosomonas and Nitrosospira, that are known AOBs that take ammonium to nitrite, and then Nitrobacter and Nitrospira that are NOBs that take it on the rest of the way. Back in the day, and in fact, I remember on the first certification test I ever took, they asked what the species of bacteria was that converted nitrite to, that did the second stage of nitrification. And the answer was Nitrobacter, because that's what all the books said. And as it turns out, Nitrobacter got all the credit for the simple reason that it was the only one that could be cultured in the lab and grown in the lab and identified by the means back in the day. It's probably back in the 70s, I would guess, or even earlier. But they could find that particular organism by conventional bacteriological identification methods, and so it got all the glory. Turns out that it's a very minor player in nitrification. Nitrospira is the principal, is the big dog on the porch. So, we live and we learn. The steps are the same, but the credit is not properly attributed and is changing as we speak. AOBs then oxidize ammonium for energy. I think this is just kind of expanding. If you're looking at for the chemistry here, ammonium, the stoichiometric ratio, the weight relationships between these, ammonium and oxygen goes to form nitrite and some water. And I think the relevant part here is it's producing a fair amount of acid. Okay. And this is one of the things that makes nitrification a little bit so troublesome, is that the acid is going to have impact on the water in which it's contained, right? And so that acid is going to react with what acids react with. Well, alkalinity is the buffering capacity of your water, right? And so what keeps your water neutral pH is the fact that it can absorb acids or bases and not have the pH swing very much. So if we've got a lot of absorptive capacity of acid, which is a code for saying a lot of alkalinity, nitrification proceeds very nicely. The acid that's produced here is readily absorbed by the alkalinity, and the pH rocks along normal. And let's talk about the football game on Friday night or something else, but there's not relevance here to this little piece of trivia. But if alkalinity is short or scarce, then a little bit of acid can take the pH quite a ways. And sometimes what we call pH porpoising, where if you're looking at your pH trend, you'll be running along and nitrifying, and the pH will just kind of go down like a porpoise is going down, and then it'll be down for a while, and then it'll kind of come back on its own. And there's a couple of reasons why that is happening. One of which is maybe the alkalinity is rebuilding itself and buffering itself back up. But the other thing that happens is if the pH gets too low, nitrification starts shutting down. And when nitrification is shutting down, they're not making acid at that level anymore, and it's going to sweeten itself back up. And so this kind of sine wave of porpoising pH oftentimes is a symptom of nitrification that's going at different rates in a system that's not well buffered. And when I say it's not well buffered, it isn't the fault of the operator that there's not enough buffer. It's a characteristic of the water that we're treating. So it's a good idea to keep some knowledge of what the alkalinity is of the wastewater that we're treating, just so that this little trivial fact that we're talking about here on the acid production is something that we can deal with if we have to. Some folks find the need then to supplement alkalinity to keep that pH up, because the nitrifiers do get sensitive to low pH, and the rate of nitrification will be adversely impacted when the pH gets down kind of in the mid sixes or below. It's kind of that's rocky road for nitrifiers to walk along. Optimum pH for nitrification is actually on the slightly neutral side, slightly alkaline side of neutral. But a lot of times we'll see these running down in the mid to upper sixes, and that's I think it's okay, but it's near an edge. All right, so that's the acid story. Nitrite oxidizing bacteria then finish the job. Nitrospira does that. You'll see in both reactions that oxygen is a component. It's a requirement there. So in terms of the oxygen costs, we're having to provide oxygen for the first step. We're having to provide oxygen for the second step. And this is where the beauty or the elegance of side stream treatments that Leon's going to talk about comes into being. Because if there's a way of shortcutting nitrification without having to go from nitrite to nitrate, the oxygen that is shown in that equation that's going to drive that is unnecessary. And that's very possible. And Leon will show you how to do that. So full aerobic nitrification is probably the highest energy cost. If we can shortcut and get there, and you're going to learn that. That's worth whatever you have to put down initially as a down payment on that, because you will get paid back over and over and over again in energy savings. Okay, so the overall equation looks like that. Just to summarize then, Nitrous ammonius and Nitrosospira are examples of ammonia oxidizing bacteria, and Nitrospira is an example of an NOB. It's only part of the story. Like I said, you'll hear more. The nitrifiers have some pretty basic characteristics, and I think everybody probably knows these by now. Obviously, they're aerobic. We've been talking so much about the oxygen requirements that they're aerobic. They're autotrophic, and remember from yesterday we used the term autotrophic and heterotrophic to illustrate differences in carbon sources. Remember, an autotrophic bacterium uses an inorganic carbon source, and heterotrophic bacteria uses an organic carbon source. The BOD removing bugs are heterotrophic. They're looking for organic carbon. Nitrifiers are autotrophic, so they're not competing for the carbon sources that the heterotrophs are. These are feeding out of it's the vegans and the meat eaters. They can go down the chow line and not get in too much conflict over the main course. Okay. Nitrifiers tend to be pretty slow growers, so that ends up being very important in setting the SRT. Remember, the SRT is the kind of the age of the biology. How long are we maintaining the average bug in that system before it's wasted? And of course, that goes to its growth rate, and the growth rate is also a function of the temperature. So not only are they slow growing, but at colder temperatures, they're even slower growing. So SRT may need to be adjusted for winter operation versus summer operation to account for both the fact that they're just naturally slow, and then they just get slower when it gets colder and faster when it gets warmer. Nitrifiers are not particularly good flock formers. Remember, we talked yesterday quite a bit about the production of EPS, the exocellular polymeric substances, the glue that holds the flock together, and the importance of good flocculation and good biopolymer is that it's the way you capture the solids. Things that grow in a suspended growth system get to a secondary clarifier where liquid solid separation is going to make or break the day. And unless it's sticky and settles, what you grow is going to wash it out. Nitrifiers like to wash out if they can't hang on to something that will help them settle. So typically we see nitrifiers retained in a system in association with sticky flocks. So there's a kind of a synergy, or at least a mutualism going on there, where there's a benefit to the company of the heterotrophs that are making the flock. It's almost like, those are the big, the heterotrophs kind of are the big lazy boy recliner, and the nitrifier goes and sits down in it and stays in a comfortable spot and travels, stays in the loop. So we they have some biopolymer. They do form some flocks, but it's not the density that we really need for really good solids capture in secondary clarifiers. Eric will be talking about secondary clarifier function later in the day, so I'll let him deal with solid separation at a much deeper depth than I'm going through here. Another important feature to remember is that the nitrifiers don't have a storage function for ammonia, contrary to what we look at for phosphorus removing biology, which basically exists and competes because it can store, right? So the ammonia removal really demands having the right retention time for that oxidation to happen, because they have to keep processing that ammonia while they're in the conditions to break it down or metabolize it. What isn't finished by the time they get out of the aeration is going to be ammonia that's passing through. Whereas phosphorus, you can see this on your phosphorus removal profiles. Phosphorus will be taken up and stored in that storage zone very, very, very quickly, and it'll travel right along with the biology through the duration of the aeration cycle. So storage is good for phosphorus. Storage doesn't happen with ammonia. It takes, stoichiometrically, one part of oxygen to oxidize part of BOD. To oxidize ammonia takes about 4.6 parts. This is, in other words, the ammonia carries a lot of oxygen punch to it. So in terms of the impact that nitrification has, it's looking for oxygen to be oxidized. Now, we've been talking all morning now about nitrification being relevant to the discussion because we're trying to get meet our effluent requirements, right? We have an ammonia limit in our effluent. We may have a total inorganic nitrogen limit in our effluent, but it's in the context of treatment. The other thing that I want you to remember is that nitrification may impact our operations simply in your BOD testing. Nitrification is just a thing that happens everywhere in nature, and we're talking about it in the context of the aeration basin. Let's go into the laboratory and run a BOD test. You run a BOD test depending on what is being put in that BOD bottle for a seed. Now, the seed is the microbial inoculum that is being introduced into that bottle that allows the BOD to reveal itself. In other words, you got to have bugs in the BOD bottle to eat the carbon that demands the oxygen to have a BOD test happen, right? So there's a microbial community in a BOD test. If there are nitrifiers in that seed, and it's very easy to have that happen, and there's ammonia in the BOD bottle, and that's very easy to happen if you've got ammonia in your effluent, you will get oxygen demand attributable to the oxidation of ammonia in addition to the BOD that is exerted by the carbon. So what I'm trying to elucidate here is the fact that in the BOD test, there's really two components that make up the BOD, and this could be helpful for certification testing, if not otherwise. If you've heard the term CBOD, CBOD is referring to the amount of biochemical oxygen demand assignable to the carbonaceous part of the wastewater, right? That's the CBOD. The total BOD is the amount of biochemical oxygen demand assignable to anything that's in the wastewater, not just the carbonaceous fraction. So anything else that's in there that can be biologically oxidized will exert demand. And ammonia is such a thing. And that's why I've got this on the screen now that say a little bit of ammonia can have a lot of BOD. One part of ammonia fully oxidized in the BOD bottle will create another 4.6 parts of, theoretically, 4.6 parts of BOD. This is a curve that was produced to show BOD results over a long period of time. What they did here is they set up a BOD test. They took a sample of wastewater and set up 21 bottles. And they checked the amount of oxygen demand at every single day over 21 days. All right? Why in the heck would you do that? Well, to reveal, maybe just for fun, but the fun is the geeky part of revealing something about the nitrogenous oxygen demand. So at time 0, there's no BOD. At the end of the first day, you've got a certain amount of BOD at the end of the second day, third day, fourth day. How many days does the standard BOD test go for? 5 days. So that oxygen demand at that 5-day point is a reportable 5-day BOD. If you keep running the bottles out, on the 6th day, would the BOD be higher than on the 5th day?
Yes.
Okay. How about on the 7th day? Yeah. The BOD keeps going up, and then something funny happens here. What we're showing is, say, after 7 or 8 days. There's another increase now in demand that's taken off. What's going on? This is more oxygen demand. What's happening here is the nitrifiers in that bottle have finally built their numbers up to where the ammonia is now being oxidized. You start with a small amount of nitrifier inoculum, and they're slow growers, but they're building their numbers up in that BOD bottle, and you may not see it at 5 days because you don't have enough of them to begin with. But if you run that test out, you'll see that the carbonaceous material, that's disappeared long ago. But the nitrogenous demand goes on for a long time. And this is, of course, why regulators are concerned about discharge of ammonia to the receiving water, because you have an infinite amount of time once it gets into the stream for the nitrogenous oxygen demand to be met. It's going to be oxygen. It's oxygen demand just as legitimate as 5-day BOD demand is, right? So I'm pointing this out because your knowledge of nitrification at some point in your career might be useful in troubleshooting poor BOD results. I'm going to show you this quick case history. This is treatment plant. Oh, so let me back up here. The way this test is done, and some of you are very familiar with this, I know, is that when you set up these bottles, these 21 bottles, you actually set up 42 bottles. In the first 21, you have just your samples. In the other 21, you have just your samples plus another chemical that's added that inhibits any nitrification. So it's what we would call the CBOD. So we can shut off any potential nitrification by adding a chemical that stops the nitrifier. And that's how this green curve is produced, is this is the inhibited, or the CBOD. And the second set of bottles then was the uninhibited that allowed the nitrifying bacteria then to replicate and to finally take off and go. Okay, so that's the difference between a total BOD and a CBOD. Is one is allowing for the total is allowing for any nitrification to be expressed because it's uninhibited. Okay, so now in this treatment plant, this is the same type of this is real live round now. The CBOD is the red, the total BOD is the yellow. This is the sample here was just the raw sewage that comes into the treatment plant. And they set these up, and the CBOD and the total BOD are basically the same thing until about day 7 or 8. And then exactly what we illustrated earlier goes on. So your nitrogenous BOD picks up. Then they go downstream from the raw influent, and they take the primary effluent, and they look at that. And they find out that, gosh, in the primary effluent, I started to get nitrogenous oxygen demand a little bit earlier. Okay. Then we go on down, and this plant has a trickling filter as their secondary treatment method. And so the effluent from the trickling filter, now remember, a trickling filter is growing a biofilm with all matter organisms on it, bugs that like carbon, bugs that like ammonia. And trickling filters are always shedding or sloughing trickling filter growth into the final effluent. And so what's happened to the onset of nitrification in the BOD bottle when we put trickling filter effluent in it? It's taken off earlier, isn't it? Why do you suppose nitrification is happening faster in the trickling filter effluent sample than it did earlier on?
How they had the bugs grow.
Yeah, because nitrifiers growing on the trickling filter were sloughing off and seeding that sample. Okay? You put the bugs in the bottle that know how to eat the ammonia, they will eat it. If you've got enough of them, they will show you that in oxygen demand. This was the kind of the coup de grace here, because this plant actually got into a violation condition for violating their total BOD. This is the final effluent. The final effluent is the red, the carbonaceous is the red line. What would you say the quality of the final effluent is from a CBOD standpoint? Doggone good, it's bragging rights. It's under 5 ppm. This could be Texas effluent. It's Oregon effluent, but it could be Texas. That good. It's not quite as big, but that's good. What's going on here? The 5-day BOD, uninhibited, was almost 25 ppm. And that's what they had to report, because they'd have to report total BOD. What's happening in the BOD bottle? Why is the reportable BOD so high? There's nitrification. Yeah, there's nitrification. They had no ammonia limit on this plant. They were not regulated on ammonia. They're regulated. They just had to keep the BOD down. Okay, how do you fix that?
Well, a couple ways, I guess. I mean, you can do it in your disinfection stage if you wanted to, or you can do it.
Because the regulator looks at this and they say, your BODs are not meeting standard. You must improve your BOD removal in your treatment plant. You're looking at a many $1 million modification of this treatment plant to solve a non-compliant BOD problem. Oh, but it's just CBOD. No, you've got a total BOD permit limit. We have no ammonia limit. Yeah, but you got a total BOD limit. Here's the clincher. When they checked their sample lines, they found out that the tubing that pulled the final sample for the composite sampler had not been changed forever. It was full of a biofilm. and the source of the nitrifiers that were being introduced into their final effluent sample because this was after the chlorine contact in that sample station, that sample line, grew a nitrifying biofilm over who knows how long, that every time that sampler pulled a sample, it scoured off nitrifiers and it was seeding nitrifiers into the final effluent sample that were an artifact of just the effluent sampling station. And for the price of tubing and changing to weekly, pull the tube, throw it in a bucket of bleach, put a new one on, and they swap their tubing out. And I think it was probably at the time, maybe $5, $10. A many $1 million upgrade did not happen because it was an artifact. It was an artifact created by nitrifiers doing what nitrifiers do, right? So, this is maybe a little bit of a side story on nitrification, but understanding the whole nitrification biology, I think, can play out in different ways. And I can tell you, this is not a one-off. I have run into this. I can think of 4, and if I think hard enough, I'll probably come up with more. 4 examples in different parts of North America that, having learned this, was exactly the same thing playing out. So it happens. So paying attention from the lab standpoint, just paying attention to the cleanliness of the sample conditions and the integrity of that whole part of it can really, it's a game changer. It really is a game changer. Yes, ma'am. Morgan.
So I'm trying to understand the impact of the receiving. So understand that they're have to test for totality.
Yeah.
And if they didn't have the organism, treat that, okay, that it's not going to show in the 5-day deal.
Right.
But isn't it ultimately
Oh,
their effluent? That's
absolutely. Oh, yeah. Ultimately, that river, this is the Willamette River in Oregon.
Yeah.
Ultimately, the Willamette River will take that ammonia load and treat it over the stretch of the river.
Okay, so it's not, I guess, the impact of.
Yeah, but the river was not, the river could handle the ammonia, because the direct, the stretch of the river and you look at the oxygen budget in that stretch of the river, there wasn't an oxygen sag that is attributable to the ammonia. So there wasn't avoidance of a problem just by the trick of getting a better sample on the final.
So they might as well just have a CBOD effluent then.
That's the other way to maybe go at this, is try to get a modification of the permit standard to a CBOD. And we've done that in a number of cases, and typically in negotiations where we usually have to give up some BOD. In other words, if they had a 30 ppm limit and you want to go with CBOD, you can get 25. 30 total will give you a 25C if you want to take that. So we have to put a little bit of BOD on the table to get that, and that might be a good trade. But I just wanted folks to understand the impact that sampling has and where this nitrification knowledge that you have fits.
So Paul, was that representative of what was going into the river though? Because that was just their sampling area, correct? So that wasn't really representative of what was going into the river though, as effluent?
It was showing you what the impact of ammonia oxidation in the BOD bottle was. Yeah, and this was actually the data that was collected during the time interval when they first looked at this, and then this is what it looked like after they went to simply weekly bleaching of the sample lines. Okay. Nitrification, denitrification then has a alkalinity requirement to be knowledgeable about. 7 parts of alkalinity gets burned up per part of ammonia fully oxidized. So that's a fairly big demand. If you've got maybe only 50 to 100 ppm of alkalinity to start with, very low buffered water, and you're going to oxidize 10 or 20 parts of ammonia, put on the spurs and grab the saddle horn, because this is going to be a rough ride. That one, we're not staying on that one very long. This is just an example then of a denitrification maneuver. This is actually from a treatment plant down at Disney World, the Reedy Creek plant, where it's an oxidation ditch, and they got into low effluent pH when it was operating in a fully aerobic mode. And then when they simply started switching off aerators to create an anoxic condition and allow denitrification to occur, you get back about half the alkalinity that is lost during nitrification, during denitrification. And so this was just a switch from aerobic to anoxic control to recover the alkalinity. So knowing that a lot of times will give you some a way to mitigate. Nitrite lock, we've already beat that one up quite a bit. You know what that is. It means that we're only getting part way, and the implications of that for disinfection have been felt long and hard here at Crude. You get there by some sort of inhibition that may be low oxygen, pH getting down below a tolerable level, getting too cold or getting toxic. I think we've seen probably the toxicity one play out most seriously, with that tetramethylammonium hydroxide hiccup, or more than a hiccup. That was a deep dive off of a long cliff, but you've got various ways to slow things down. I've already shown you this relationship of 5 to 1. You may see this in certification tests. A lot of demand of chlorine for the nitrite. You've had nitrite lock here. Optimum pH on the slightly alkaline side, minimum down about 6, in the mid 6s, max up around 10. This is illustrating the idea that as temperature cools off, of course, growth rate slows down, and if we're trying to nitrify, then we have to make adjustments in the SRT to accordingly. We've said that now about 10 times. Denitrification, you've seen this graphic previously, showing you again. So the nitrate's coming back to nitrogen gas. You call that clumping, pin flock ashing, deflocculation, or turbidity. What we're illustrating up here on the front end is a denitrification simulation. We're going to get here to the break here in just a couple of minutes, and we'll take a look at that example. We're simply reducing then the nitrate to nitrogen gas that was produced, the nitrate that was produced during aerobic treatment. In this case, we're showing a carbon source. See this one carbon, OH makes it an alcohol, one carbon alcohol, that's methanol. Very common single carbon alcohol that's used to provide a carbon source for denitrification. In our simulation here, we do have one of the test vessels with some methanol in it. I used an alternative carbon source as well, so we're testing 2 different carbon sources. But basically, they're using the oxidation power of the nitrate to metabolize or oxidize that carbon source and produce the nitrogen gas and give you back the alkalinity. The denitrification biology is much simpler than nitrification biology because a lot of organisms have the ability to denitrify. They're very common in wastewater. They're facultative and heterotrophic. Facultative meaning that they swing, through the whole range of oxidation ORPs, and they're heterotrophic, meaning they use organic carbon. This is probably looks maybe a little bit confusing. What I want to simply illustrate is that when we talked yesterday about the metabolic sequence that produces energy to the cell, that there was this thing called an electron transport chain, and there's a set of metabolic pathways. There's very well-known and documented stepwise sequence by which, as electrons are taken out and moved along to finally deliver energy to the cell, they go through a bunch of steps in a very predictable sequence. And at the terminal end, there has to be something that receives or accepts those electrons at the tail end, a final hydrogen acceptor. And what I'm illustrating here is kind of part of the metabolic sequence that you would see if you look at a biochemistry book. And you don't need to know much about this graphic other than the fact that at the very tail end of electron transport, when these electrons are being handed off, the final resting place, this is where oxygen sits. Remember, oxygen for an aerobe then is saying, give me, I'm the terminal acceptor. I'm going to be here and receive that. And oxygen gets reduced to water, okay? This bacterium then that's going to denitrify simply puts nitrate right into that last step. It slips right into the same slot. And so you reduce nitrate to nitrogen gas, and it happens almost within a heartbeat. It's a very, very quick shift from aerobic electron transport to anoxic electron transport. All right. So I just wanted to see kind of where the nitrate fits logically right in as a terminal, what we say a terminal electron acceptor. The benefits of denitrification, I think, are probably pretty obvious. It's removing the nitrogen now from the water and putting it into the atmosphere from which it came. And it's doing treatment. Remember, we're using it as oxidizing power to do treatment. So it's removing DOD and giving us back some alkalinity. So denitrification, I think we should probably sing the praises of more than we would do. It reduces energy of use, of course, then to the degree that the oxidizing power of that is returned to us, and puts the nitrogen into a place where it doesn't do harm. Nitrate's a strong oxidizer. We get almost 3 pounds of BOD oxidized per pound of nitrate reduced. So you've seen these relationships, and now you can see where the nitrate fits in there. So lots to like about that. The alkalinity that's produced is about half of what was consumed. The impacts of denitrification usually are felt in solid separation issues, secondary clarifier, where there's a tendency for the sludge maybe to deflocculate a bit or to rise and create TSS. What we're trying to illustrate here then is denitrification that does occur simultaneously just because of the biodynamics of the site of the flocks and the aeration strategy that you happen to be utilizing. If we've got flocks that are large enough and good enough aeration control, we actually then see oxygen gradient that gets low as we move through a flock with an anoxic center to the flock. And anytime you get anoxic, then you have the potential then for the nitrate to actually be denitrified right within the flock. So you saw this example yesterday. You're seeing it again. Now, I think this is a repeat of a slide that I showed yesterday, but this is actually the beautiful Cruz granular flock now with the ammonia, nitrate. This is nitrification. We're in anoxic conditions, and the denitrifiers are converting it off to nitrogen gas. And that's simply going to strip out of the aeration basin. I think Mike was saying maybe 50 or 60% of the denitrification is actually occurring within the aeration basin by simultaneous denitrification. Concurrent with that, in the same culture, in the same flock, of course, is your heterotrophic DOD removal is going on side by side. So the flock is doing a lot of things, and we're not even showing anything about phosphorus going on in that slide. So it's a busy Grand Central Station where transformation of nutrients is occurring. To denitrify, they have to be there. They are. It's hard not to have them there, but we have to get the DO down very, very low, because the bugs, remember, on that electron tower will take what gives them the best value. And oxygen is more attractive from an energy yield standpoint. So if we can get the DO very close to zero, then it forces them to switch over and stick nitrate in there at the tail end. And it certainly helps to have a reason to be needing an oxidizer, which is having a carbon source. So there's always a low rate of denitrification that's driven in the absence of an exogenous carbon source because of the endogenous respiration rate. You know, the bugs continue to respire even though they've eaten all the BOD. There's still a metabolic rate that we call endogenous. So endo means inside, genus body. So endogenous means respiration driven by what's inside the body. If we have food on the outside, carbon, that's exogenous. Exo is exit or outside. So exogenous respiration is at a higher rate. And when we're adding carbon sources, then we have exogenous respiratory acceleration, if you will, that is over the endogenous. But even a complete BOD removable sludge is still potentially going to denitrify given enough time, just because of the endogenous rate. The bugs keep oxidizing even when they're asleep, and hopefully we do too, because we like to wake up in the morning. This is simulate. This is what we've tried to set up here now in our simulation. This is the cruise system. We've picked up samples of what we call aerator influent, and by aerator influent, what we're referring to is samples collected right at the head end of the reactor. Remember, the flow's going this way. So we've got flow coming into the aeration basin that should have unmetabolized carbon. It should have a fair amount of ammonia. It likely won't have much nitrate because it hasn't oxidized the ammonia until it gets downstream, right? So the first two samples were collected from this spot. The last four samples were collected from the back end. And so those are the same bugs, but now they've had an opportunity to eat the BOD, to oxidize the ammonia. And what we've done then is I've said, okay, the first one is simply going to be a control. It's the bugs as they're entering that aeration cycle. They got BOD on board, they got ammonia on board, they probably don't have much nitrate on board unless somehow it came back around with them. The second one, what I've added to it is some nitrate. I said, okay, we're going to give you some nitrate, and you've got a carbon source that you carried on with you, because that's in the primary effluent. So that's what's in the second figure. The next one was simply saying, all right, let's see how you're doing when we take you out at the back end. So we would expect at the back end, the BOD ought to be pretty well gone. The nitrate should be produced. And we'll see what the potential is for that to denitrify. You know, you got some nitrate there. Is there enough carbon to drive much denitrification? I don't know. This next one, let's see, what did I put in there? Okay. I said, all right, we think you got nitrate because you had to make it, but I'm going to make sure you got a lot of nitrate, so I'm going to add some to you. So we've simply really boosted the nitrate in that 1, 2, 3, 4. Yeah, I can still count to 4. The fourth sample. Then we've got the same sample, and to this one I added a carbon source plus nitrate. So I boosted the nitrate and I gave it some carbon source. And the carbon source I put in here was acetate. Acetate's very simple, readily degradable simple carbon. And then for the last one, I said, okay, we're going to add the nitrate and we're going to give it an alternative carbon source. We're going to give it the one that a lot of folks have been using for years. We're going to give it some ethanol. Okay, and the whole idea was to say, all right, knowing what we put in there, you know, what would you predict would be the first one to go? And of course, you know what the first one to go is because you saw it. It was the sample, I believe it was this one, and that was the sample in which you had nitrate, and we added acetate. That must have really liked the acetate. It that was, mainline and acetate into the veins of these little buggers, and they had the nitrate there, and they made a lot of gas. So that one went first. The second one to go was this primary, was the one that had carbon from the, whatever's coming in from the primary effluent. So it's carrying its raw BOD. It's BOD that it knows it likes because it's what it always gets, right? So it's a good carbon source. And all it needed was some nitrate, away she went. So that would make sense. What would you say the third one what's the most likely next one to go? And we'll see.
The last one.
Probably the last one, because we know it's got the carbon source, it's got the nitrate. Kind of a surprise it hasn't gone yet, but I think it's, it doesn't read my book and give a rip about what I say, so it's going to do what it does. What will be the very last or the least likely to go?
The aerator, effluent, I think.
The aerator,
The effluent,
the aerator effluent. So the aerator effluent would have no carbon, but what does the aerator effluent also have?
nitrate.
Its own nitrate that it made.
Yeah.
Okay. This one has carbon because it has its BOD but it has not made any nitrate yet. And denitrification is being driven by nitrate reduction, right? So does it make sense that this one, if these boogers read the book, this one ought to go last, or probably never?
Yeah, that makes sense.
Does that make sense?
Yeah.
Okay. You have more than